Bonjour, je m'appelle Anca Apavaloaei. J'ai étudié à l'IRIC à l'Université de Montréal dans le laboratoire de Dr. Claude Perreault. Le titre de ma thèse est Exploring the Landscape of Actionable HLA-Associated Tumor Antigens Across Cancers. The key to developing safe and effective cancer immunotherapies is finding targetable antigens that are specific to cancer, present in cancer, but absent from normal tissues. Most mutations do not generate targetable antigens. And when they do, these mutations and the tumor antigens from those mutations are patient-specific, and the therapies cannot be scaled at the population level. The search for tumor antigens has been focused on the 2% of the genome coding for classical proteins and ignored the other 98% that was previously considered junk DNA. So this approach of predicting tumor antigens is very restrictive and has a high error rate. Instead of predicting antigens, we developed a strategy to directly sequence the tumor antigens from patients' tumors. And then we traced the origin of those tumor antigens back into the genome. We were surprised to find that most tumor antigens derive from the so-called junk DNA, and they were not mutated. In other words, most tumor antigens derive from genomic regions that are the same in normal and cancer cells, but they are activated specifically in cancer. These antigens then expand the pool of targetable antigens that we can use for cancer immunotherapies, and they would have been completely ignored with the standard predictive approaches. Because these antigens are not mutated but hard-coded in our genome, they have a high likelihood of being shared between patients. In fact, every tumor type that we have studied in the lab presented groups of shared antigens characteristic to that particular cancer type. And this allows us to develop off-the-shelf vaccines with antigens that are most prevalent in one particular cancer type. And even more, we found a unique set of tumor antigens that were shared between multiple cancer types. And this set of tumor antigens was associated with aggressive tumor features, but can also form the basis of a universal anti-cancer vaccine. Once an antigen has been found, there are numerous methods to target it. Our favorite approach is to train the patient's own immune system to recognize these antigens using vaccines. These can be mRNA vaccines that have now been proven safe and effective, and that allow to encode multiple antigens that are shared across one cancer or multiple cancer types into one shot. And our lab is hopeful that we can start clinical trials using these antigens in less than 2 years.